at Oxford, both students and postdocs who've worked in my lab over the last 15 years, and some of the people are named here. And so why are we interested in the tropics? Lots of reasons, of biodiversity, in terms of many key ecosystem functions that the tropics provide. But in terms of the climate change, perhaps the most relevant part of the tropical biosphere is its role in the global carbon cycle, and how it absorbs carbon and releases carbon to the atmosphere. And if you look in terms of productivity, the total photosynthesis of the land surface, you can see that the wet tropics in particular, but the drier tropics as well, dominate in terms of global terrestrial productivity. And here you see it in red across the three main tropical forest zones. And we understand why that is. It's the year-long growing season, the lack of a dry season or a winter season of dormancy that causes this very high level of productivity. And if we look at it in another way, this is a work that one of the advantages of being in a geography department is that I work with a lot of colleagues who think a lot about maps. And another way of looking at this feature is by this type of map, which is called a cartogram, where the size of each pixel is related to the variable. So you see your familiar tropical forest map with all of the continents arrayed there, but now you see a distortion because this is giving you a map of the biological size of the land surface in terms of biological activity, total photosynthesis. And what we see here is that on the annual, when you sum over the whole year, the tropics are bulged out compared to your normal geographic map, and the dry regions, the Sahara Desert and other areas shrink to almost nothing because they're very low biological productivity. If you do this in January, when the Northern Hemisphere is largely in winter and shut down, you see even more that in January, in the Northern Hemisphere winter months, we really live in a tropical planet in terms of biological activity on the land surface. And if you look at Colombia here on the left, it's about the same size as the entire, all of North America in terms of its biological activity in January. If you go to July, you see things are a bit more equally distributed. The Northern Hemisphere has greened up. The days are very long. And you see a pattern that's still distorted, but overall shows some slightly more familiar patterns overall in biological activity. And then if you do this over the full annual cycle, you see this great seasonal flux of variation in the pulse of the tropics, very strongly driven by the continuous biological activity of the tropical forests. So I'm getting a bit of interference. I don't know whether somebody can mute the microphone, just to clarify that, or maybe whoever's in control can mute people's microphones.
Sí, Verónica puede controlar los micrófonos si no tengo acceso. Edwin.
Sí, señor.
González, por favor.
Ya mismo.
Gracias. Sorry.
Okay, great. That's better. Thank you. And so, one of the questions is when we see from satellites that those maps that I showed you of productivity are very much based on simple satellite imagery, MODIS, etc. And when we look at the tropics simply in terms of the visible spectrum, we see a fairly uniform green vision of the tropical forest biome. And we can be tempted to think that most tropical forests, perhaps they work in more or less a similar fashion, that there's ultimately the productivity is driven by sunshine and rainfall, and there isn't that much variation. But I think that is a mistake. As much as it is in temperate regions, when you actually get down to understand the ecology of each particular tropical location, you often find a lot of difference. Some of it driven by environment, climate or soils, other things driven by biogeographical history, accidents of history, and possibly other factors, faunal interactions, trophic interactions that make tropical forests very different from each other. And so to partially understand this variation across the tropics, over the last 15 years or so, we created a network of intensive monitoring sites that look at the carbon dynamics of the forest, and beyond that, look at many aspects of ecosystem functioning in various parts of the tropics. And initially, this started in South America, working in the Amazon and the Andes, and then over the last decade, we've also worked a lot in Africa and in Southeast Asia, and also in some areas outside of the tropics in this work. And I'll describe briefly some of the methods that we use. Some more details are given in this manual that is available online for anyone to look at in more detail and to replicate some of these methods if they wish. Just to explain some of the concepts that we'll be using when we're describing the carbon cycle in a forest, initially the absorption of carbon dioxide from the atmosphere combined with energy to make photosynthate is the gross primary production, or the canopy photosynthesis. Of that photosynthesis, much of that captured energy, is used by the plants themselves for their own biological activity of growth, of maintenance, and that's termed the plant respiration or the autotrophic respiration. And it can be from the leaves, from the wood, or from the root system below ground. The rest of that material ends up making biomass, or that's that photosynthate, and that can be biomass in the woods, which we're perhaps most familiar with in forestry, but also it can be the leaves, the flowers, the fruit, or it can be below ground in the root system as well. And that manufacture of biomass is termed the net primary productivity. So it's the difference between the photosynthesis and the plant respiration is the net primary productivity. And all of that biomass eventually ends up as dead biomass. Most of it is when, through seasonal cycles of litter fall, when leaves or flowers or fruit are shed, or when there's root turnover and dead roots are shed, or eventually when a plant dies and the entire tree or plant becomes dead material. That is then decomposed by soil fauna, but also particularly by the microbial part of the soil, the fungi and the bacteria, and that consume that material and release CO2 back to the atmosphere as a waste product of that process. And that process is termed the heterotrophic respiration. Now, very rarely considered in these carbon cycle calculations is that some of that energy passes, and captured carbon passes into herbivores, directly, whether insects or vertebrates, consuming the plant material, and some of that then passes to higher of predation. So the later part of this talk, I'm also going to talk a little bit about some of those trophic ecology to this broad carbon cycle description of forests. And so in the GEM protocol, we've come up with a set of measurements, generally quite low cost overall, but involving regular visits to the field to collect this sort of data. We deliberately didn't want to have very complicated equipment like eddy covariance towers or complicated gas analyzers, because we wanted to make this something that could be replicated in many sites across the tropics. So we measure tree growth either through forest inventories or through very cheap homemade dendrometers that measure the growth with often monthly precision, but at least annual precision overall. We collect the canopy litter fall and separate out into flowers and fruits and leaves and twigs, and that's done typically every 2 weeks in the field. We also measure root productivity, and the most common method we use is in-growth cores, where we put in, we take out the roots from the soil and put root-free soil into these mesh bags, and then come back 3 months later and see how many roots have grown into the mesh bag, and to repeat this every 3 months to get the below ground productivity. We use other methods as well. We often use screens in the ground and to try and describe the root dynamics at a finer time scale. And then to measure carbon dioxide fluxes, we use a gas analyzer. This is perhaps the one more expensive bit of equipment we have. And with that, we measure the release of carbon dioxide from the soil by putting this chamber on the soil and seeing the rate at which carbon dioxide concentration builds up in that chamber. And we do experiments. We may have soil without litter, with roots, without roots, to try and understand how much of the carbon is coming from the roots, from the soil organic material, or from the leaf litter. And we do similar measurements of the woody stem to see how much carbon dioxide is coming out of the woody biomass in the system. And then not so regularly, but maybe once in the wet season, once in the dry season, we may do a campaign where we cut branches and look at the photosynthetic and respiration properties of the leaves as well to get the carbon cycle of those described. And then we also have weather stations and try and look at the relate the patterns we see to the climate patterns at the site. And this is what we end up with in the end, this sort of diagram. This is for a site in Peru that we did, one of the earliest sites we did in Madre de Dios in Peru. But now we have diagrams like this now, probably for around 50 or 60 sites across the tropics now, describing in detail the flow of carbon through a system. And I won't go into detail through this diagram, but just to highlight a few things. If you look at the top circle, this is the gross primary productivity, the total amount of energy flowing. Can you hear me? Okay, so that was the total amount of carbon dioxide flowing through the system, and the woody, and whereas the middle, this term in the middle is the woody production, the increase in woody biomass, which is the most of the biomass of the forest in the long term. And what you see is that the woody production is only around 7% of the total photosynthesis. So if we're trying to understand woody growth rates, and from, say, for example, remote sensing of photosynthesis, we have to realize that a woody part of the forest is only a small part of the internal carbon cycle of an ecosystem. And similarly, vice versa, if we're measuring tree growth rates or looking at tree rings and trying to say something about ecosystem productivity, just from the tree rings or the tree growth rates, we have to realize that the connection is very indirect. There's lots of other priorities that a forest is doing in terms of allocating to leaves and roots and to its own respiration that can distort that simple relationship between woody growth and canopy photosynthesis. And also at many of these sites, as well as this carbon cycle work, we try and couple this with understanding the traits of the forest canopy. So we collect leaf samples and we do chemical traits, morphological traits, and also leaf gas exchange measurements on those as well, ultimately to try and connect the canopy properties to the carbon cycle properties that we see, and ultimately perhaps to link to remote sensing approaches. And just to give you a very quick tour of some of the sites that we worked with, this work began in Amazonia originally around 2005, where we had a project trying to compare the forests of Eastern Amazonia on very infertile soils with the forests of Western Amazonia in Peru on more fertile soils, and try and understand the difference in carbon dynamics that we had there. Then a few years later, we expanded the methodology, going up from the lowland Amazon up the slope of the Andes. So this is looking at, we're standing here in this image at 3,000 meters, looking down the eastern slope of the Andes into the Amazon forest at in Madre de Dios, and we set up plots roughly every 250 meters in elevation down this gradient to try and understand how temperature and ecosystem function varied along the elevation gradient. Since around 2011, we've done similar work in Southeast Asia, in the tall dipterocarp forests of Borneo, and I'll talk a bit more about them later in my talk. And also, I've really prioritized doing a lot of work in Africa, where until this work began, there's much less tropical forest ecology, ecosystem ecology work done in African rainforests than in Amazonian forests, despite them being the second most important tropical forest region in terms of area of the world. And a lot of this work also involves capacity strengthening, working with local teams, getting many students trained up in the methods, both in their own, studying in their own home countries, but also many of them then going either to Oxford or to other international partners and universities are being trained up there. And so we've had a strong capacity strengthening, long-term commitment in many of these projects. This is a site when we got our first data from Africa in 2016. We had a workshop there to work out these data, and there's those 6 bars in the center here were the first descriptions of primary productivity in an African rainforest. So our colleagues in Gabon were very excited. A month later, our colleagues in Ghana caught up, and we had a workshop there. And at that stage, we had even more data from Africa, from zero, a few months before than we had from Amazonia or from Southeast Asia. So gradually, we were building up data sets from around the tropics and seeing how similar or different they were. And we've had other departures since then. A recent focus the last few years has also been moving out of rainforests into savannas and doing work. We had to extend the protocol to include more on the herbaceous layer, productivity, and traits, as well as the tree layers as well, to expand the savanna methodology. And now we have around 30 sites in savannas, as well as the tropical forest sites, most of them in Africa, but also in the Cerrado of Brazil. And so now moving on to some of the results. There's lots of things I could tell you in the results that we don't have time in a 45-minute seminar. So I'm just going to focus on a few of the insights that have emerged from this network and some of the new directions that we're heading with this network. So one thing we can look at is how similar is tropical forest productivity across the regions. When we looked at the start, at that MODIS-derived image of tropical forest productivity, that's very much a satellite product that produces that. How much does that reflect the ground-up measurements of what the productivity of lowland tropical forests would be? And so what we're focusing on is what I'll focus on particularly is the net primary productivity and the gross primary productivity across regions, and the ratio of the two. The ratio of net primary productivity to gross primary productivity is sometimes called the carbon use efficiency. What fraction of photosynthesis ends up producing biomass, whether it's in leaves or in woods or in fine roots. And we want to see how consistent is that carbon use efficiency across forests. So that involves understanding the respiration and the primary productivity. And I'll show you some of the work we've done comparing two regions. So Amazonia, going from wet to dry. So in these maps, the darker areas are wetter areas, the lighter areas are dry. And so in Amazonia, we had a transect in Western Amazonia on relatively fertile soils from Alpuayo near Iquitos down to a site in Bolivia, which is right at the edge of the Amazon forest biome. And then in Eastern Amazonia, on less fertile soils, we went from wet forest in Cachoeira down to very dry transition forest in Tanguru. And so in West Africa, we had a gradient going from rainforest in the southwest at Ankasa down to dry forest and semi-deciduous forest, and then dry forest savanna transition in the center of Ghana. So I wanted to compare these two regions and see what we found. And this shows you some of the results. This is work that's just been submitted by my student, Huang Zhenzhen. And so when we first did this work in Amazonia and published a few years ago, and so what we've done here in the x-axis is rank the sites from wet to dry in terms of rainfall, but also soil moisture holding capacity. And when we look at Amazonia, which is the red line here, what we found was quite a steady prediction decline. As you got drier, the productivity declined because of the dry season of leaves shedding or photosynthesis shutting down. And productivity overall was quite a predictable function of dryness. And so what we wanted to test is whether this pattern held in West Africa in the forest, and also whether the productivity was at the same level as our Amazonian sites. But we didn't find a very strong soil fertility effect. Both the fertile and infertile sites had a similar productivity. And the blue line shows our data from our West African transect. And what we found is that the wet rainforests in West Africa were quite similar to the wet forests in Amazonia in overall productivity. But when we went to the semi-deciduous forests and into the dry forests, we found much higher productivity in the West African forests than in the equivalent Amazonian forests. And when we looked at that was in terms of photosynthesis. When we looked in terms of biomass production, we still found that difference, but it was a bit less, the contrast, than the contrast in photosynthesis. And the reason was that the carbon use efficiency was higher in Amazonia. So of all the photosynthesis occurring in the Amazonian forests, more of it went to produce biomass than it did in West Africa. And we still don't fully understand why that is, but it shows, what this result shows us is that the difference in metabolism between... I can hear a voice there.
Sorry. Yeah, already.
Okay, thank you.
Okay.
And so there are clearly differences in function between these West African forests and these Amazonian forests that we're still beginning to try and understand. But what we did find, both in West Africa and more recently in Borneo, is that the productivity we measure on the ground is much higher than the MODIS predicted productivity, which is often calibrated off Amazonia, because that's where most of the data 10, 15 years ago for tropical forest function ecology was based in the neotropics. And we're finding some surprises in the Afrotropics in some areas and in Borneo of higher productivity that isn't captured in our satellite data. So the tropics overall might be more productive than we estimate simply from our current satellite-based understanding. And I have some theories about what that's happened, why that is happening, and we can discuss that if we have time in the discussion. So that tells us about the initial capture of carbon dioxide and productivity. Another question we can ask with the GEM network is, how is that allocated? So if we have this biomass productivity, does the same amount go to woody production in every site, or does it vary between sites? And if it does vary, what are the factors that are determining that variation? And so what we're looking at now is this net primary productivity, but these three components: the canopy productivity, the woody productivity, and the coarse root productivity. And we're trying to understand the relative partitioning in that productivity and how consistent or variable it is across sites. And so what we're doing in terms of measurements is comparing the canopy litter fall with the woody growth with the below ground root dynamics and seeing how they're varying. And the easiest way to show this is in a ternary diagram. Sometimes this takes a moment to get your head into these diagrams to understand them. But what broadly, the bottom axis shows the percentage of allocation of productivity to the canopy. So if a forest entirely allocated everything to canopy, it would be in the bottom right. If the bottom left is the allocation to the fine roots, with this scale on the left-hand side indicating that, and the top is the allocation to the stem or the woody productivity. And the first thing to notice is that there's quite a lot of scatter between different plots. And what we see, so what we see overall is that there's quite a lot of variation, that there isn't a consistent pattern. It isn't as if all forests have the same partitioning of resources between these three areas. And the other thing that stands out in this image is some biogeographical factor, that the Asian sites, which are all from Borneo, are all on the right-hand side. And so they're allocating much less to roots overall, and much more, and then relatively more to the other components. So, and we consistently find that in these dipterocarp forests of Asia, of Southeast Asia, and it may be linked to the ectomycorrhizal associations they have, which are different from the arbuscular mycorrhizal associations other sites have. So there is a hint of a biogeographical difference in this allocation. And so then we next try to model and predict, can we work out from climate or soil variables what factors shape this allocation? And we tried a variety of things, and it's quite noisy data, lots of factors there. But one thing that does stand out in our data is that, surprisingly, we can predict the amount of productivity allocated to roots as quite a strong function of the soil texture. The Asian sites are slightly offset from the other sites, but broadly, the sandier the soil, the more allocation there is to root production across continents, across soil types. As long as they're sandy, they have more allocation. That's a very consistent result we're seeing in these data. And the more that is allocated to roots, the less than is allocated to the canopy or to wood. So you see those terms as a forest gets sandier, those terms go down, and the allocation to the roots goes up. So we are seeing some patterns in there linked to soil texture rather than to climate or other factors. And so that gives us some potential to scale using soils data or soils maps to a larger scale. The next thing we want to do with this work is to go from the individual sites to start saying something about the functioning of tropical forests at scale. And to do that, we need to link our detailed field measurements with the spectral signatures of those forest plots, and then see whether we can infer something about the forest carbon cycle from the leaf spectral properties and calibrate it off our gem sites, and then be able to extrapolate to a larger scale. And we use multispectral approaches, where we use around between 8 and 10 bands of information in the infrared and in the visible, initially sometimes from field measurements, sometimes from drone measurements, and then ultimately we connect up to satellite data. And the different wavelengths, when you look beyond, particularly when you look beyond the visible wavelengths into the near infrared, different species with different chemistries, different leaf structures have different reflectance patterns in the infrared wavelengths that seem to be quite strongly linked to many leaf functional traits, such as leaf mass per area, leaf nitrogen content, either directly or through correlations to the leaf economic spectrum. We seem to be able to detect many leaf traits from multispectral data. And so what we do with our gem plots is sometimes using drones as an example by colleague Jesus with our multispectral drone. He's going to come to Colombia next week and fly over a variety of sites in Colombia with this instrumentation. And so what we have in the plots is the precise location where we've sampled leaf traits and the canopy properties that we've sampled in our plots. And then based on that, we look at other species of the other trees of the same species in the plot and allocate the same traits to those other trees. And then we try and link this to our drone data and our satellite data to try and see whether we can extrapolate and map those traits at larger scales, and then test that by validation, cross-validation of the data. And then we use Sentinel data to try and link to these larger scales. And so we end up, so this is work that's currently in review, we end up with attempts at mapping plant traits calibrated off our gem plots across the entire tropics. And this is work in progress. So these maps, we think they're relatively credible, but I think as we get more data, they will become more credible over time. But so these first maps are almost like our null hypothesis of our first hypothesis of what we predict plant traits to be based on our plots. And then we can, as new data comes in, we can test and improve them. So if you look across the lowland tropics, this is a specific leaf area here as a trait. So the area of leaf per gram of leaf is the inverse of the leaf mass per area. And you see that we're seeing some patterns in the lowlands that are there, some patterns in the drier forests, such as in southern China or in the Caatinga of eastern Brazil. But also we see in the montane forests, lower specific leaf area or higher leaf mass per area in the montane forests. And in the bottom one, we zoom into our transect in Peru and see this is our predicted map of how we expect leaf traits to vary in this area, Andes to Amazon, wet humid forest transition in Peru. Here's another example of a map which shows more variation. This is the leaf phosphorus content, which we don't think we can directly detect phosphorus in the multispectral data, but because of its correlation to traits that we can detect, like leaf mass per area, like leaf photosynthesis, we think that overall we're beginning to pick up patterns in phosphorus. So you see Eastern Amazonia, the infertile soils having much lower leaf phosphorus content than the sites in Western Amazonia. You see similar patterns in Africa, and overall you see Southeast Asia having generally intermediary phosphorus content, and there are very few very infertile sites, but very few fertile sites as well. So we're starting to see interesting patterns across the continents that we need to test and validate. But for the first time, we can start mapping traits at a pantropical level that can ultimately inform our ecosystem models. So we're not treating the tropics as this homogeneous green sameness, but actually start getting some of that spatial variation in tropical forest function into our models. And here's another one. This is leaf photosynthetic traits, VC max, leaf carboxylation rate. And again, we're trying to see, we're seeing interesting variations and patterns across the tropics. And so the final thing I want to do is now go beyond the carbon cycle and to think about the final bits that I showed in that diagram, how much of the energy captured by the forest ends up being consumed by higher trophic levels in the forest. And so in this diagram, we're going to look at these two arrows, which, as I said, we hadn't previously, until the last year or two, really looked at in terms of tropical forest ecology and ecosystem and dynamics, the herbivory and the predation. And the work we're going to do is focused on Southeast Asia and these magnificent tall dipterocarp forests of Southeast Asia, typical canopy height around 50 meters in a magnificent tall old growth forests, but also very heavily logged sites as well. So these forests have a lot of timber species per hectare, so the selective logging is very intensive. There are typically 40 trees per hectare taken out. So there's a very intense logging and structurally degraded forests. And so these forests are often termed degraded forests in terms of when we think of red plus or in terms of carbon content. And that can be a useful thing if it stimulates restoration or of these forests, but also in many contexts, this labeling these forests degraded is also a bit dangerous because it encourages further conversion to oil palm plantations or to other agricultural uses that companies or governments say, we're not going to deforest anymore, we're going to leave our old growth forests, but we're going to just work on degraded lands or degraded forests, and we're going to convert them because they already have lost a lot of their value. It doesn't matter so much if they're converted into agriculture. And I want to show that actually that assumption of degradation is a bit dangerous. So if we look in terms of biomass and forest structure, it's undoubtedly the case that old growth forests having a magnificent tall forest canopies, 60 meters average, as you can see these LiDAR scans, whereas the heavily logged forests, you've lost that structure, that height, you've got a very much denser understory, and you've lost all the big trees in these forests. So structurally, they clearly are heavily logged and degraded. And in terms of biomass, we see that as well in terms of carbon stocks. The old growth forests, the logged forests have about half the carbon stocks of the old growth forest, and then the oil palm plantations have even less. And that's including the soil. If you don't include the soil, it's in much greater reduction. So undoubtedly, there's quite a lot of loss of carbon associated with this logging. When we look at productivity using our GEM protocol, we see not much change in productivity, maybe a slight increase in the logged forest compared to the old growth forest, and high productivity in the oil palm, but most of that is in the oil palm fruit that are taken out of the system overall. And so just like I've done at the other sites, we can produce these GEM style diagrams. And this is one that compares the old growth forest in terms of carbon cycling to the heavily logged forests. And one thing you notice, the green numbers are for the old growth forest, so brown numbers are for the logged forest. And so in terms of net carbon balance, or NEE, you see that the old growth forests are near carbon neutral. Maybe they're a carbon sink with a negative number, but it's not a significantly different from zero with this methodology. but the logged forests are clearly a carbon source. So these are the forests that were logged 20 years ago. So this is not what we were expecting. We were expecting the biomass to be increasing strongly in these forests. And so even though the initial logging released carbon, we expected the forests to be now absorbing lots of carbon as they increase in biomass. But that's not what we measured in these forests. And we had both flux towers and gen measurements. And where is this extra carbon coming from? A lot of it's coming from the dead wood. So there's still a lot of dead woody debris in these forests. So that's releasing much more carbon from the logged forest than compared to the old growth forest. We see that the woody growth is indeed much higher in the logged forest. It is almost twice as high as these trees are racing for the light, trying to close those gaps. They're prioritizing woody growth. But that doesn't offset the extra release from the dead wood. But also we see that the other significant thing is we're seeing more carbon being released from the soil in the logged forest than in the old growth forest. And so there seems to be more biological activity in the soil, maybe priming with all the material in there. The soil microbes seem to be digging into the soil carbon and releasing more soil carbon to the atmosphere. So in terms of carbon balance, the impact of logging was actually worse and longer lived than we expected. That even 20 years after logging, this forest was releasing carbon to the atmosphere. And also this showed the importance of considering the whole ecosystem. If we were just doing classic forestry and just going in and measuring the woody biomass and reporting that as the carbon balance of this forest, we would decide this forest is a big carbon sink now post-logging. But actually, once you look at the whole system, it is still quite a strong carbon source. And so so in terms of carbon, these forests were worse than we expected. But how are they in terms of ecological function? And the approach that we took to look at ecological function was to think about energy and the flow of energy through the forest. Now, the carbon cycle, everything is the flow of captured sunshine. So the carbon cycle numbers can easily be converted into energy flow simply by a simple multiplying factor linked to the energy content of photosynthate. And the reason that we're using energy here rather than carbon is that it can work across all the taxa. It's a physically meaningful unit that can be consistently applied to plants, but also to animals as well, and reflects metabolic function and activity of organisms. And so the question we wanted to ask is how much of this ecosystem productivity ends up going through, in this case, key vertebrates, the birds and mammals. In principle, you could do this for invertebrates, for other things, but some of the best data you often find are mammal abundances and bird abundances. And so we focused on those to look at this question. And why is energy interesting? Because we argue that the energy flow through particular gills, whether seed dispersers or pollinators or nutrient cyclers or predators, can be a proxy for the strength of that ecosystem function, that ecosystem service in that ecosystem. And so for the sites in Borneo, we were lucky because as well as our carbon cycle work, this was part of a much larger program of research. And our colleagues at the University of Canterbury had collected a lot of detailed data on mammal population densities and bird population densities that we could relate to. So if we knew the population density, we then calculated from body mass the energy requirement of each species, and then we could use that to compare that and see how much energy is needed to support that population density that we see there. And so we found a number of surprises when we did this. One is that to our surprise, the energy flow in the logged forest through the birds, so essentially it's the population density of birds, but converted into energy units, was 2.5 times greater in the logged forest than in the old growth forest before collapsing back in the oil palm. And the same story was found in the mammals. There were more mammals consuming more energy in the logged forest than in the old growth. And also that could happen, but it could be that maybe that's just a few generalist species taking over the forest. the date is wrong though. This paper was last year, no, 2022. I just noticed I typed in 2002 on the slide there. So this is the paper that came out a few months ago. And what we found is that the species diversity, the number of species contributing to the energy flow, the S numbers in these diagrams, didn't decline in the logged forest. So it wasn't just as if a few generalists were taking over. It was a high diversity of species contributing to the energy flow in each of the gills. Whether you can see the arboreal insectivorous birds here, the size of the circle is the amount of energy flow. You can see the terrestrial herbivores here, quite small in the logged forest, quite a large number, quite small in the old growth forest, quite a large number in the logged forest before everything collapses in the oil palm plantation. So we're seeing lots of diversity in each of these gills supporting the energy flow. And so this could also still be negative. It could be that the old growth specialist species are declining and disturbance adapted species are taking over. So what we did is look at the individual species and how the energy flow in each of those species changed over time. And here I'm just showing the top 20 species in terms of the winners and the bottom 20 species in terms of losers. And there's a lot of species in between these ones, but you can see from the split that most of them will have been winners, because even the 20th one is still showing quite a strong increase in energy flow. So what we found overall, the deer and the elephants were the biggest winners. So many of the birds, the generalist birds, bullbills did well, but most old growth forest species increased in abundance in the logged forest, and only three old growth forest species showed significant declines: 2 spiny rats and a mouse deer in abundance. And generally, most of the old growth forest species increased in abundance in the logged forest, which is quite a surprise. We didn't expect this. So why are these logged forests having amplified energetics? And basically, we think it comes down to bottom-up factors, that as the plants are prioritizing rapid growth, they're investing less in chemical defense and more in rapid growth. And so their leaves are more palatable, less chemically defended. That's increasing the insect and arthropod herbivory that supports the insectivores. There's also there's more vegetation near the ground that's more palatable and tasty. So the terrestrial large herbivores and omnivores also have more food. So the entire forest becomes less chemically defended and more palatable or salad-like overall. And there may also be some top-down factors in terms of predation, protection from predation, but it's really, we think it's the bottom-up factors that are dominating. So this is one point. So I want to be hesitant in saying whether this is a general pattern. We need to replicate this sort of approach in other sites to see whether the same pattern holds. And there may be biogeographical or other factors that mean the same story isn't true in the neotropics or the afrotropics. So I have to have some caution in this result overall. But it does show that how we could extend the GEM approach to go beyond just thinking about vegetation, to start including a holistic view that includes the animal communities as well, and get a more holistic idea of ecological function by using this energetic view of forests. So to conclude, this GEM global network is giving new insights into the productivity and allocation of carbon in tropical forests. And what we're finding is that different tropical forests have different relationships between productivity and environment. So the tropics aren't all the same. They're very different in functioning. And then just the last few things, going beyond the vegetation to try to have a more holistic view of the functioning of a whole forest. The amount of energy flow flowing through the higher trophic levels, we argue, is perhaps a measure of the ecological vibrancy of an ecosystem. Not just the amount, but also the diversity of species contributing to that energy flow. And at least at the Borneo sites, the structurally disturbed forests, to our surprise, appear to be more ecologically vibrant than the old growth forests. At least in terms of birds and mammals. We haven't looked at the invertebrates there. And so the carbon and biodiversity stories are going in different directions. The logging is worse for carbon than we expected, but it is not as harmful for biodiversity as we expected, at least in this study site. So thank you. So I'll stop there.
Okay.
Thanks a lot, Yadvinder, perfectly on time. So I leave the word to Veronica, which is coordinating the question and answer part of the seminar.
Okay,
Veronica?
thank you so. Okay, thank you so much. for your presentation, Dr. Malhi. It's been a pleasure and an honor to have you with us at the research seminary, and I apologize to you, Professor Nicola, and attendants, because I had serious technical problems at the beginning of the seminar. So we have a few minutes for questions, and please raise your hand to participate, or you can write in the chat directly, preferably in English. Okay, let me see. Professor Posada, por favor.
Hi, Yadvinder, thank you very much. That was a great seminar. I have a bunch of questions, but I'm going to hold to a few. One is about the increase, the comparison that you did between Africa and the neotropics with the GPP, that it was even along this drought gradient that it stayed higher in Africa.
Yes.
So I have a vague memory of reading a paper in Africa that along these drought gradients, there was a difference because the cloudiness was higher in those forests. So it was dry, it was maybe dry, it was dry, there was less precipitation, but at the end there was also less evapotranspiration. Yes. Because of an increased cloudiness. Have you looked into that? Do you think that could explain why GPP stays higher?
That's an interesting suggestion. Yeah, no, we haven't looked at it in this context, but it is true. Less so in Ghana, but particularly in Gabon and Central Africa, you have the dry season is cloudy, the entire dry season. So you can support forests at quite low rainfall. In the neotropics, you would certainly have savanna in those conditions. So it's possible that it's a factor, and I hadn't thought about that. So that's a good suggestion. My other theory around this is that actually where those West African forests get very heavy dust deposition from the Sahara, and we hear a lot about Amazonia getting Saharan dust, but West Africa gets hundreds of times more dust from the Sahara than Amazonia does. So, and that dust from the Sahara is mainly from dried up lake beds, so it's mainly dried up fish bone, so it's phosphorus and calcium. So I think these forests are also constantly limed, essentially, and their pH is around 7 rather than around 4. And so there may also be this interesting interaction with the Saharan dust that's also driving this high productivity.
Great. Can I ask a quick question? A second quick question?
Okay.
I also was curious about why do you think the NPP measure in the field gives higher values than when it's estimated from the MODIS sensors? Do you have a...
Yeah, I actually, my student Juan Yan has just been looking into this very closely. Firstly, the MODIS NPP product, if you look at it, a lot of it is just default values because it saturates in tropical conditions. So you're not actually seeing a lot of variation in the MODIS signal. And then what we find, when we look closely, is that the cloud contamination is still an issue in the MODIS signal. It's meant to be removed, but it's actually very hard to remove it in a wet tropical area as well. And so that drives down the estimated productivity, the seasonality. Apparently MODIS sees seasonality, but actually it's cloud contamination that it's seeing. And we've done a nice little study in West Africa showing that. Yeah, I think why MODIS tends to agree in Eastern Amazonia is, I think, initially when it was being calibrated, the only data that were available were from the LBA project in Brazil. And so it's not that things were fitted to that, but it was that you stop, once your model predictions and start matching your field data, you stop playing around with your model to think it's okay. So I think there's a bias where MODIS fits well to where the data existed 10, 15 years ago, which is Amazonia. And when we're applying it across the tropics, we're actually seeing there's a lot of divergence between MODIS and the field data.
Okay.
Thanks.
Thank you so much. Professor Stein, por favor.
Hey, Vinder, thanks for a nice talk. Yeah, I have also a couple of questions, but one was about the deposition, and in fact I wanted to ask something like there. I mainly work on forest fires, so in Central Africa I was observed from all the savanna fires, they have these really huge nitrogen depositions. I wondered of course the Sahara dust is also there where it passes over where you have your forest plots, but I wondered if you have been thinking about the forest fire aerosol depositions as well.
Yeah, that's an interesting question. I think there's less in the West Africa. There's less of that forest, that aerosol deposition signal, just the way the air currents flow. But it could be part of the signal. It's possible that there is something there around the forest fires as well.
Yeah, and then my real question was like one of the things mainly about like the human appropriation of MPP. So like you have like less MPP which can be used by like other species, of course. And
Yes.
so like the data about your energy fluxes like in locked forest was really surprising. I wondered like in a like selective locked like Amazonian forest, for example, would you expect something similar? Because those are first they're very heavily locked, right? And so if you have like more selectively locked, like a couple of trees removed, would you expect something similar? And that there's like in fact like a higher flux towards like other parts of life from MPP, or don't you think so?
Yeah, I think my guess is that for the neotropics you'll see a similar pattern, but obviously with the lighter the logging, there's probably some sort of a pumped relationship that a certain amount of logging enhances energy flow, and then once you've really started degrading the system, then the energy flow cuts off. So I think my expectation is that we would see a similar pattern in the neotropics. We're actually currently writing a grant to try and replicate that sort of work in Brazil, and try and see whether we do see similar patterns or not there. But I think the basic print mechanism to me is fairly intuitive. And actually, when after that paper came out, when I've talked to various people, people like ornithologists, bird people, they say actually this is roughly what we see when you look at a disturbed forest or log forest. You often see more birds of the old growth, of the forest birds as well there, but it hasn't been quantified in the same way. So I think a lot of people intuitively do sense that moderate disturbance seems to enhance animal populations, which is linked to the intermediate disturbance hypothesis that's been around for a long time. But this is a quantifiable way of perhaps identifying the process.
Yeah, I was going to go towards the hypothesis. So do you think in fact that like, although like humans are like the human appropriation of MPP is like really high, in fact like the disturbance is like offsetting that partially?
In this particular context, yeah, I think and I think we have to be careful. And I should emphasize one thing all that work is showing is I'm not saying we should go and log all the old growth forests. There's lots of value in the old growth forests for lots of reasons, and lots we don't know or understand as well. So this is not an advocacy for logging everything. But what it does seem to suggest is that old growth systems are in a very chemically conservative state, heavily chemically defended, low palatability, and then some disturbance of that system can shift that into a more palatable state that does boost many animal populations in that system. So there does seem to, and the mechanisms seem quite reasonable, quite plausible. Why that works.
Yeah. And do you expect like from natural disturbances that something similar is happening?
Absolutely, I think so. Certainly, blowdowns or other things like that.
Okay, very nice. Thanks.
Thank you.
Natalia, por favor.
Thank you very much for the presentation. I was wondering about when you compare the Asia, America, and Africa in terms of the ectomycorrhizae and arbuscular mycorrhizae. How is that comparison, or whether you expected to compare, and the second question is whether you take into account the trees that you analyze where ectomycorrhizal tree or arbuscular.
Yeah, so the measurements of the roots were done at plot level, so we didn't distinguish which species was the root productivity, but those Borneo forests are dominated by dipterocarp trees that are ectomycorrhizal. So we expect the tree root community to be predominantly ectomycorrhizal in the Borneo sites and not in the other sites. And so what I didn't go into detail there is when we measure root productivity, we're just looking at what's allocated to root biomass. What we're not capturing there is what's being allocated to mycorrhizae. So what I think is happening in the Asian forests is that the below ground carbon allocation may be the same, but more of it's going into mycorrhizal systems that are doing the foraging, and less is going into building new roots, because the roots don't need to forage so much because the mycorrhizae are doing doing the foraging very efficiently. So maybe the shift is between how much of your below ground carbon you allocate to plant roots and how much you allocate to the exomycorrhizae. And I think what's happening in Asia, we haven't proved this yet, but my feeling is that what's happening in those Borneo forests is much more is being allocated to the ectomycorrhizae. And why? I mean, it's still a bit of a mystery why dipterocarps dominate in Southeast Asia. There's various theories, but no one really has a final convincing reason why those forests are dipterocarp dominated.
Yeah, but you take also dipterocarpaceae from the tropics, well, from the Amazon part, or not?
We haven't got any. I know there are a few dipterocarps in Colombia. I know there are some in Africa, but we haven't really had plots in those dipterocarp forests. Well, I think the Amazonian one is just individual trees. They don't dominate in a forest in the way they do in Borneo.
Thank you.
Thank you.
Profesor Posada, para terminar.
Yeah,
Por favor.
just I'm gonna, since I don't see more raised hands, I will ask another question. Have you looked at or it's much harder to measure, but have you looked at the allocation to reproduction along those different gradients, like ecosystem level?
Yes, we have, in the sense that we've with our litter fall, we separate out into fruit and flowers and leaves, and so in places like the Andean transect, my colleague Cécile Girardin has published some stories, examination of the allocation to reproduction. What we haven't done yet, and I think it's an obvious thing to do, is look pantropically, to look at that, what the story is in allocation to reproduction and how it varies between regions. So that's an interesting question. That we have the data, but we have no one. I'll get a student to look at that, I think, at some point.
Thanks.
Professor Stein, por favor. Professor Stein.
Yeah, but as no one else is, I have another question, because you have all these studies on biodiversity or species richness and productivity, and on average, African forests tend to be species poorer than Amazonia, but your productivity is higher. So what is going I wonder if you have been thinking that direction as well. Yeah.
My feeling is that those productivity function relationships, I think, most of the literature is from grasslands and things. I think they only hold when you have quite low diversity. So maybe if you're going from 1 species per hectare to 5 species per hectare or 10 species per hectare, you might see these interactions between diversity and productivity. But I think in most tropical forests, once you start going above 20 species per hectare, I think the signal's lost. I think I don't expect diversity to link to productivity at all. And without analyzing it specifically, we see no evidence of that in our data. We don't see the more diverse forests as being more productive beyond what climate can explain. Dry season and those things matter, but beyond that, we don't see. But diversity seems to add any extra information in terms of productivity.
Yeah. And do you have a guess why the African forests are more productive when they are slightly drier than the very wettest sites?
Yeah, so not all African forests. So the data from Gabon, which I didn't show, it seems to be similar to Brazilian Amazonia, and it's on a similar old substrates or Guyana. So it is only some African forests, and I think that there are 3 possibilities. That 2 we've already discussed. One is possibly deposition. They're much more basic overall because of that deposition. They're like pH 7. So the soil ecology is different. The one Juan mentioned, maybe it's to do with the cloudiness, so that for the same dryness, they actually are not as water stressed as Amazonian forests there. And the third one might also be to do with longer term disturbance history. These forests, they're mature forests, they're maybe over 100, 150 years old, but they were disturbed a lot in the 19th century, linked to the slave trade and various things that rural areas were depopulated. Many of these forests recovered from agricultural systems when people fled the rural areas to urban areas because of slavery. And so there may be a long-term legacy of of impact there, that these forests are still, it's more than 100 years, but there may still be a human disturbance recovery signal there that's much stronger than for example, in Amazonia, where the pre-Columbian impact and things is much further back in time.
Okay, thanks a lot.
Okay, let me see. Okay, I don't see other questions or hand raises. Dr. Malhi, thanks for your time, and thanks for accepting our invitation, and to the participants for joining us and for staying active and interested in the faculty research seminar. So good afternoon to all. Thank you so much.
Thank you very much. It's been a pleasure. Really
Thanks a lot, Yadvinder. Sorry for the time.
No problem.
In your case, already past dinner time. Thanks again.
Thank you.
Thank you so
Yeah.
much.
Bye.